everyone, everything. A very good afternoon, ladies and gentlemen. It is my pleasure to welcome everyone to the webinar on Bioinformatics Concept and Development, which is organized by KKR College of Agricultural Biotechnology, Nashik. We are so excited to have you all here as a participant. Nowadays, we all are stuck at our home due to the pandemic situation. But learning from new is our passion. So we all are here to crack the knowledge about the bioinformatics, which is nothing but the combination of the computer science and research areas of the biotechnology or biology field. So now I heartily welcome to Honorable Mr. Wagh, President of Wagh Education Society, Nashik, for the valuable presence. I welcome to all the trustees, secretaries of Wagh Education Society for this online webinar. I heartily welcome to our today's resource person, Mr. Ashish Wani, Professor at Biotechnology Department, Arts, Commerce and Science College, Nagar. Teachers are some of the most unsung heroes we have. And I feel so happy to say that one sir is my teacher. He always inspires us to do something new. Then I welcome Dr. V.M. Sherika sir, Campus Coordinator, Wagh Education Campus, Saraswati Nagar, Panchvati, Nashik. I also welcome Professor N.S. Kajpur sir, Principal of Wagh College of Agricultural Biotechnology, Nashik. Also, I heartily welcome to all the dignitaries, principals, HODs, teachers, and student participants to this webinar. I request Dr. N.S. Kajpur sir for giving introduction about our Wagh Education Society.
Thank you, madam. Good afternoon, all of you. So, firstly, I am welcome Professor Ashish Wani sir, Professor Biotechnology Department, New Art, Commerce and Science College, Nagar. So, this is our 5 online lecture webinar, and I am proud that the Professor Ashish Wani end this webinar session with this important topic on this bioinformatics, that is concept and the development. And on behalf of Wagh College of Agriculture, Biotechnology and Wagh Education Society, I welcome Professor Ashish Wani sir. So before the start of our webinar, so I am give the brief information about the Wagh Education Society. So Wagh Education Society is established in 1970 by the Padma Shri Karmavir Kakasaheb Wagh. It is the one of the most important personality in this 1898 to 1983 era, and the education is the one of the need of this social thing. So that's why they are started in 1970 the Wagh English School or Wagh Vidya Bhavan at Bhausaheb Nagar. And from this beginning, now in days, this Wagh Education Society, they are give the education in different areas, which include the engineering college, MBA, MCA, Polytechnic, Pharmacy, Agri and Agri-allied colleges, senior colleges, fine arts, performing arts, nursing, B.Ed, junior colleges, CBSE, and the different state and state board schools. So in 1970, starting from the 1970, now in 2020, so Wagh Education Society is they are celebrating the 50 years of their establishment. And now until today, we have a 36 different institutes, which in which the near about 20,000 students they are studying, and we are providing these the services to these 20,000 students with our faculty and the total staff near about 2,000 staff. They are providing the servicing to this number of students. Then important milestones in the Wagh Education Society development. So as in 1970 Wagh Vidya Bhavan is started, And with this start, the Kekewak Education Society, they are entered into the technical education in 1983 with this Kekewak start of Kekewak College of Polytechnic. Then in 1984, Kekewak Institute of Engineering and Education. Now this engineering college is one of the most branded and NAC accredited A grade college in the Maharashtra. Then in 2003, the Kekewak Education Society entered into the agriculture education. So now under this Kekewak Education Society, six different agriculture branches they are now running. The one of the our branch, that is agriculture biotechnology, started in 2008. And with this start in 2018, recently we started the Kekewak Institute of Pharmacy at Chandori and at Nashik. So in different areas of education, so at the Kekewak education they are entered and providing the, that is the quality knowledge, and become a one of the excellence center for the education in the Nasik and in the Maharashtra. Then the different institute which are started by this Kekewak Education Society. With this, we are having the polytechnics. Then we are having the art, commerce, science colleges, and then we are having the six agriculture and agriculture allied colleges. Next. Then under this situation of COVID-19, so we are using the different technologies. So these technologies include the, that is different e-resources. Then we are providing the assignments to the students, online assignments. Then under this COVID-19 situation, the connection between the teacher and students, we are establishing by creating the different videos on the various topics of the subject, and providing we are published on this YouTube channel. And this education, because of this COVID-19 situation, we cannot stop the education. We are continuously in touch with the student by using these different technologies. And these different technologies, they are also very helpful for to communicate the students and to evaluate the students by using the different softwares. Then this is about the our recruiters. So after the completion of education, different institutes, they are inviting the different multinational companies and important companies from the India's for to recruit our students, and we provide the placement to the students in different multinational companies which include the Tata, then ABA. These are the different industries which come to our campus and take this placement drive to recruit the student in their in very that is the particularly in their industries. And then with the one of the our objective is that after the completion of degree, we must connect with the our alumni. So we organize different alumni meets in India and in abroad also. So at Washington DC, we organized this alumni meet in 2016, and then at St. Joe's in USA. Then at recently in 2019 at Houston, Texas, we organized this alumni meet. And with the celebration of 50 years of establishment, we organized one grand alumni meet at our engineering college on 21st December 2019. And near about 10,000 students, they are attending this grand alumni meet. Then these are the intake of this our polytechnics. So we are having this four polytechnics at different regions with near about intake capacity of 1,350. Then beside this, we are having the Kekewak Nursing, Kekewak Education, Fine Arts, Performing Arts, and the Pharmacy College. Then the that is traditional college, that is the BSc, BCom, and BA. So we are having this different in the village region, that is at Chandori, Kakasaheb Nagar. In agriculture, in rural areas, we also providing the education to the student by opening these colleges at village level. Then in agriculture education, we having six agriculture colleges. This is the one of the example of education society which having these six colleges under the one education society under one roof. So which are these agriculture engineering, agriculture, agricultural biotechnology, food technology, agriculture business management, and KKO College of Horticulture. So near about 440 with intake, we provide providing the education in different 6 branches of agriculture. Then these are some our initiatives of this KKWAG Education Society in order to reach to the society and reach to the students. That is Guru Shishya Punyatithi competitions. So this competition is for particularly for the school students. So to develop their different characteristics at the school level education. So we organize these Guru Shishya Punyatithi competitions. Then KKWAG Kala Academy, so not only the education in the traditional, we also provide the education in fine arts and performing arts, and we organize the program related to this, which develop this interest of the students in different these areas. So that's why we are organized the program of KKWAG Kala Academy. Then Karmavir Smruti Sachak competition. So this competition is particularly for the rural areas, this particularly the students which are in the rural areas, and we are having some sports ambitions. So that's why we are organizing this competition at the village level in order to promote these students, and to organize this competition and to connect with the students which are having these sports ambitions. Then public lecture series. So likewise, the not only in education, so there are some different social issues are there. So we organize the public lecture series for all this public. They are open for all the public in different topics which are related to our social needs. Then Karma Yogi and Krushi Tapasvi Puraskar. So Karma Yogi Puraskar is related for the social work. So there are different people, they are working in various areas for the social work. So we are not nominate, we are not called the application for this Puraskar. We choose and we identify the students who are working in isolated for the social purpose. So we give this Puraskar Karma Yogi and Krushi Tapasvi for the outstanding work in agriculture sector. Then convocation at society level. So we organize this convocation under the Pune University. Then important, that is the CEO meet and the HR connect. So recruitment of student is based on the industry need. So what is the industry and what is the need of industry, and how these students they are developed in this the industry needs. So in order to that, for this better communication, we organize CEO meet and HR connect at our institute. And important, that is the science project competition to include the research that is interest in the students. So that's why we organize this science project competition. So these are the our initiatives for the to connect with the society and to connect with the students. So this is the brief information of this society. Thank you for listening me. So once again, I am welcome Professor Ashish Wani sir for this today's webinar on the important topics, and near about from the different areas of these students. They are now participating in this webinar. So I think this is the very useful webinar for the agriculture biotechnology point of view, and for the students and the faculty and other members who are interested in the areas of bioinformatics. Thank you.
Thank you so much, sir, for such a wonderful presentation. Now, I would like to request Professor C.S. Nirgude, sir, to give the introduction of our college.
First of all, very good afternoon to all of you which are present here, all delegates, researchers, students, faculties from the various fields on the behalf of KKWAG College of Agriculture Biotechnology. I am C.S. Nirgude to here for sharing some features of our college, that is KKWAG College of Agriculture Biotechnology here. So why we choose, why you pursue the agriculture education in the KKWAG? One of the most important point is that this is the this organization, that is the KKWAG Education Society. It is from last 15 years working in the education sector. That means KKWAG is one of the most important brand in the education sector. And in the case of agriculture, here we have 6 different branches of agriculture and allied colleges. That is agriculture, engineering is there, agriculture is there, biotechnology is there, horticulture is there, food technology is there, and agriculture business management. These 6 different departments or different colleges are in one roof. So this is the one of the most important point in all Maharashtra, in all Maharashtra states. There is only one society that pursue these 6 different type of particular courses in the field of agriculture. And one of the most important point is that these all 6 colleges is affiliated to the MPKV Rahuri, which is the one of the most famous agriculture in whole India. After that, in our college, there is a various sessions is there organized by the our institute. One of the event is that is the Krishi Udyog Sangam, which is organized by our institute with the collaboration with the CII on the 21 to 22 September 2017. So in this, our Vice Chancellor of MPKV Rauri, Dr. KP Vishwanatha, is addressing us as a chief guest. After that, why agriculture biotechnology? So all of you know that agriculture biotechnology is the multidisciplinary science. It involves the plant biotechnology, molecular biology, immunology, microbiology, animal biotechnology, and other fields also. One of the in that bioinformatics is one of the most important factor or department in the agriculture biotechnology. Then the scope and opportunities of for the agriculture biotechnology is that we can become agriculture research scientist through the ASRB board, then the researcher, independent researcher, then the various research associate posts which are filled in the particular various research institute, then the technical staff for the R&D and R&D, that is the research and development in the National Institute, quality manager. These are the various scopes for the agriculture biotechnology in the particular life science field. After that, laboratory facilities which are available in our college, there is a 7 different departments or laboratories there, and which well furnished, and it have a various types of instrument, just like PCR, fermenter, gel documentation. After that, we comes to our student profile. From the last 9 years, our institute 100% seats are get filled of our institute. Then the highest cutoff than any other non-aided private institute in the case of biotechnology, and almost 338 students were graduated till 1819. After that, our students, our college students are here actively participate in various activities, just like research competitions, sports, conferences, and poster presentation. Among that, academic excellence, Archana Navle in 1920 received Best Agriculture Student Award. After that, Saurabh Gorpode has worked as a trainee in the Indian Institute of Management, Ahmedabad. In the case of in the sector of sports, Mr. Ganesh Narsale received the silver medal in the grappling, which is held at the Rohtak. And in poster and conference, is Mr. Ashutosh Shetty in 1819 got a first prize in the MPKV Rahuri. Then the well qualified experienced staff is there. Total 15 teaching staff is there, and non-teaching is 34. Then the our college is organized some interaction with the our faculty and the particular student with the other areas lectures or persons. For that, expert lecture is organized by our institute. Then the industrial visit is organized, and hands-on training, that is the HOT for the research purpose. HOT is run by our institute. After that, success rate. This is the success rate of our alumni in the various fields. Then these are our distinguished alumni. Within that, one of the remarkable or milestone is that Mr. Sagar Yadav pursuing which the PhD in the biological science from the CSIR National Chemical Laboratory NCL Pune. Then Vishal Zagade, which is the PSI at Nashik. Then the Harshal Sonone is a agriculture officer in SBI, that is the banking sector. There is a lot of students of our institute that are working in the banking sector. And then the Harshala Galan is the working as a Krishi Sevak in the Igatpuri region. Then the Rohan Shradonkar, which is a PhD in the biological sciences in Northern Illinois University, higher study for the in abroad. Then the Trishala Pagar, which is the technical training in the Dr. Reddy's laboratory. So this is the somewhat our features of our college. Thank you for this presentation.
Now, I would like to request the same Professor C.S. to give a brief introduction of our research.
Thank you, ma'am. It's my pleasure to introduce our today's resource person, Professor Ashish Wani. He has completed his graduation and post-graduation in the field of biotechnology in the North Maharashtra University, Jalgaon in 2009. Sir has cracked GATE in life science in 2009 only, and also he cleared CSIR NET life in the field of life science in 2010 and he also cleared SET in the life science field exam in 2011. He has specialization in molecular biology, genetic engineering, animal biotechnology, bioinformatics. He worked as a junior research fellow in ISER Pune during 2009 and 10. He's worked on two different innovative projects. He is currently working as assistant professor in New Arts Commerce Science College, Ahmednagar, and as a examiner, paper setter, moderator in the Pune University. He has his own 3 research publications, 6 synopses. Also, he organized 3 different workshops like ICR, CSIR NET, SET guidance, HPLC technique in molecular biology. Also, he participated in 7 different national conferences. Also, he currently has worked on his own coaching for competitive exams like NET, SET, GATE, DVT, JRF. I feel so glad to introduce such a multitasking personality. Thank you, sir, for your accepting our request.
Thank you so much. Now, I would like to request Professor Ashish Wani sir to initiate their lecture. So please.
Yeah, thank you, sir. Thank you for such a generous introduction. And I would like to congratulate Wagh Education Society as well as Wagh College of Agriculture for the great work they are doing in the field of education. And I would like to thank Principal of Wagh College of Agriculture, Professor Passport sir, then Coordinator of Saraswati Nagar Campus, Sevlikar sir, and Webinar Coordinator, Professor Kulkarni Madam, for inviting me for this webinar. So today's webinar topic, as you know, it is bioinformatics concept and development. So as you know, in the last century was there was a tremendous development in the field of computer sciences, and now the new technology in the field of computer sciences is artificial intelligence and this social networking, etc., etc. Like in the field of biology or in the field of biotechnology, the new avenue for the development is bioinformatics. So every biotechnologist or biologist should have the knowledge of bioinformatics for the to sustain in this field and to achieve their future goals. So as you all know that in modern biology, these quantitation tools are very important. We have to measure something in our day-to-day life, in our practical work, in our experiment work, and we use variety of tools or instruments for that purpose. Like we use pH meter for the pH measurement, we use spectrophotometer for the measurement of the optical density, and after measurement, we perform the quantitative analysis of that data which is generated. Now, Usually we perform this quantitative analysis by using a graph paper or a pen and paper, because the number of data sets, they are very minimum in our routine practical work or experiment work. Like take the example of your biochemistry practical, that is finding out the concentration of protein or sugar in the unknown solution. For that purpose, you plot the standard curve and then you find out the concentration of the unknown. But just imagine if you have hundreds of readings or hundreds of data points, then it is absolutely difficult for a human being to quantitatively analyze that data. So in that case, a very simple quantitative tool that we use for the analysis is Microsoft Excel. Similarly, nowadays in the field of biological sciences, enormous amount of data is being produced, and that data has to be analyzed with the help of computer. And there comes bioinformatics into the picture, because with the help of bioinformatics, we can store this enormous amount of data, as well as we can analyze this enormous amount of data with the help of quantitative tools. So this field of bioinformatics is due to the development in two major fields, that is molecular biology and computer science, over the past 30 to 40 years. The first protein sequence database, which was established in 1965, it is called as Atlas of Protein Sequence and Structure, established by Margaret Dayhoff. Then in 1970, Brookhaven National Laboratory established Protein Data Bank for archiving three-dimensional protein structures. So you must know that these 3D protein structures are determined by X-ray crystallography or NMR technique. And these three-dimensional structures, they are stored in a specialized data called as a database called as Protein Data Bank. Earlier, in this protein data bank, less than a dozen of structure were present, but now the size of this protein data bank has been increased enormously, and more than 1 lakh structures are stored in the protein data bank today. These are some historical milestones in the field of bioinformatics. That is, in 1970, first algorithm for the global alignment was written by Needleman and Wunsch. Then in 1974, Chow and Fasman, they write the algorithm for secondary structure prediction of the proteins. And in 1980s, there was establishment of a database called as GenBank by NCBI. And later on, FASTA and BLAST, these two quantitative tools for database similarity searching were developed. And in 1980s, the Human Genome Project was started. And this Human Genome Project was give a major boost for the development of bioinformatics. And later on, in 1990s, there was a widespread use of internet. And due to that, again, this bioinformatics was spread across the globe. From DNA to genome. Now, this development in the field of molecular biology and bioinformatics goes hand in hand. As we progresses into the field of molecular biology or genomics, simultaneously there is a progress in the field of bioinformatics. Like in 1953, the first DNA model was developed by Watson and Crick, the double helix model. And in 1955, Sanger sequenced the insulin protein. And again in 1978, Sanger dideoxyDNA sequencing method was developed by Frederick Sanger. So Frederick Sanger is the person who received the Nobel Prize twice. First of all, he received for sequencing the insulin protein, and later he received the Nobel Prize for dideoxy sequencing method development. Later on, in 1985, Carey Mullis developed invent the process of PCR, that is polymerase chain reaction. Now, similarly, as you can see, there is a development in the field of molecular biology. At the same time, there were developments in the field of bioinformatics were taking place. So as soon as we come to know the method of protein sequence determination, structure determination, the first database was developed by Margaret Dayhoff called as Atlas of Protein Sequences and Structure to store protein sequences and structure. Then ARPANET, actually this is the early internet, which was developed by US Army for intercommunication. But later on, it became the WWW, that is World Wide Web, that is internet. Then in 1973, PDB, that is Protein Data Bank, was established to store the protein structures. And again in 1982, GenBank was established by NCBI, which is a nucleotide sequence database where the sequences of DNA and RNA are stored. Later on, in late 1980s, Human Genome Project was initiated. And in 1995, the first bacterial genome was completely sequenced. The bacteria was Haemophilus influenzae. And in 1996, the whole yeast genome was sequenced. Later on, in 2001, the first human genome draft was published. And you know that in 2003, we come to know about the whole genome sequence. Now, similarly, in late 1980s, Swiss Prot database was established, and the Swiss Prot database was specifically dedicated for protein sequences storage. Later on, there was establishment of NCBI, and a tool was developed called as FASTA. Now, FASTA was developed by William Pearson, and it is a database similarity search tool. And later on in 1990, BLAST tool was developed, that is Basic Local Alignment Search Tool. And this tool is again a database similarity search tool developed by Stephen Earl School at NCBI. And in 1991, WWW, that is World Wide Web, that is Internet, was developed. And later on, again, it gave the major boost for the development of bioinformatics. And then in 1992, EBI, that is European Bioinformatics Institute, was established in the Europe for the development of bioinformatics. So as I told you that this field, these two fields, molecular biology and bioinformatics, goes hand in hand. The major reason behind is that in late 1970s, two methods for DNA sequencing were developed. One was developed by Frederick Sanger, and other was developed by Maxam and Gilbert method. Now, both of these methods, they determine the DNA sequence by performing the page at the end of the experiment, that is polyacrylamide gel electrophoresis. And due to this whole cumbersome process, only few hundreds of nucleotide sequence was determined after the strenuous exercise. But in 1984, first automated DNA sequencer was developed at Applied Biosciences, which automized the Sanger's dideoxy method of DNA sequencing. And when this automated DNA sequencers come into picture, many of the workers started to work on the DNA molecule. Earlier to that, we know in the history of biology, people initially work at the organism level, then they started to work at the organ level, then tissue level. When we invent the microscope, then people move on the cellular level. And then we come to know about the molecules. So now we are working at the molecular level. So in order to understand or in order to determine the DNA sequence, that how a gene function, how it responds to the external stimulus, we have to know about the DNA sequence of that gene. So this automated sequencers make that task very easy for us. And many laboratories, they procure this instrument and they started to work on the DNA or protein level. Now in this graph, you can see after 1994, there is an exponential increase in the number of sequences which were determined, as well as number of base pairs of DNA which were determined by a sequencing instrument. Now, this is old data. In 2008 itself, almost 100 billion base pair of DNA sequence data was already known, and almost 100 million of DNA sequences we come to know. And now the situation is that if you try to write all these DNA sequences on the paper, the whole paper in the world will not be sufficient to write all these DNA sequence data. Now, when we talk about automated sequencers. Nowadays, next generation sequencing platforms are available, like Illumina sequencing is there, or Roche 454 sequencers are there, which are based on pyro sequencing technique, or there were this earlier applied biosystems sequencing instrument, which were based on the Sanger's method. They are also present, as well as the ion torrent sequencing instruments are now available. Now, by using this next generation sequencing platform, you can determine the whole genome sequence within a tangible time frame. So, as you can see, this is the genome size of some organisms, like a very simple virus, T2 bacteriophage. It has a genome size of around 1,70,000 bases. A simple bacteria, which is a model organism of the prokaryotic system, E. coli, its genome size is around 4.6 millions of base pairs. Then again, a model organism of insects, Drosophila melanogaster, its genome size is 130 million base pairs. Then Homo sapiens, our genome size is around 3.2 billions of bases. So when we say 3.2 billions of bases, it roughly around 320 crores of base pairs. So these many base pairs are present in just a single cell haploid chromosome. Then Paris japonica, which is a canopy plant, around 150 billions of base pairs. Now in this graph, you can see as the complexity of organism increases, the genome size also increases. Like in for case of viruses, the genome size is very small, roughly around 10 kb to 100 kb. For bacteria, 1 MB to 10 MB. MB stands for mega base pairs, also called as millions of base pairs. And for higher eukaryotes, you can see for plants, fishes, mammals, reptiles, the genome size roughly ranging in between 1 GB, that is 1 gigabase pair, it is also called as 1 billions of base pair, to 100 billions of base pair. So when you determine the whole genome sequences of these organisms, enormous amount of DNA sequence data is actually produced. Up till now, we have these many numbers of complete genome sequences already determined, like 37 eukaryotic whole genome sequences are determined, and almost 1,708 whole genome sequence of prokaryotes have been already determined by using this next generation sequencing platforms. So what we do in this field of bioinformatics, when with the help of this sequencing instrument, we generate this sequence data. Apparently, it is a very complex set of data. Now, with the help of bioinformatics, we convert that complex data into the knowledge. So the whole understanding of these genomic sequences, like take an example, like suppose you have a whole genome sequence of a human. Now the next task is you have to find out where the genes are present. Now again, with the help of bioinformatics tools, we can find out the genic region, or where the genes are present. Next, we can, with the help of bioinformatics tool, we can figure out what is the function of this gene, and what kind of protein products they are produced, what kind of different proteins they are interacting with each other. So this whole load of knowledge we can generate by analyzing this complex data. But as I mentioned, this genomic sequence data is very large in size. It is impossible for a human being to analyze it manually. We take help of bioinformatics tools, quantitative tools or computational tools, for analysis of this data and for the knowledge generation. So as I already mentioned, that enormous amount of biological data is being produced due to development in the field of molecular biology, automated sequencing. Now, demand for efficient computational tools to manage and analyze this data was increased, and there this bioinformatics came into picture. So this development of computational tools, it depends on knowledge generated from wide range of disciplines. So that's why bioinformatics is a multidisciplinary field where you have to have knowledge of mathematics, statistics, computer science, information technology, and molecular biology, of course. And merger of these all disciplines, it created an information-oriented field in biology, which is known as bioinformatics. So you can see the bioinformatics is at the center of all these different disciplines of the sciences. It touches all aspects of science: engineering, computer science, chemistry, biochemistry, biology, statistics, and mathematics. So what is bioinformatics? I will tell you a very simple definition of bioinformatics. You just have to remember 4 words and you will always remember the definition of bioinformatics. So bioinformatics is an interdisciplinary research area at the interface between computer science and biological science. And it can be defined as union of biology and informatics. Bioinformatics involve the technology that uses computers for storage, retrieval, manipulation, and distribution of information related to biological macromolecules such as DNA, RNA, and protein. So all these 4 words comprises the whole definition of bioinformatics: storage, retrieval, manipulation, and distribution. I can explain it with the help of a very simple example. Nowadays, whole bunch of data that we store in computers or in our mobile phones. Our mobile phones are also small computers. They have processor, they have RAM, they have input and output devices. So everything, it may be a photograph, it may be a song, it may be a movie, you store it into the your computer or in your mobile phones. Similarly, the genomic sequence data or the whole sequence data of DNA or proteins, we need to save it into such a system which has ability to store enormous amount of data, and that is computer. Next thing is that whenever we save the data, we always get it back from the computer, right? That is called as retrieval. So again, with the help of retrieval, again with the help of computers, we can retrieve the DNA sequence data or protein sequence data from the computers. Third thing that we do with the use of computer is analysis. All sort of data you analyze with the help of computer. Again, this sequence data or the structured data we can analyze with the help of computational tools or softwares in the computer. And finally, today, the medium of distribution of data is again internet. Now, internet is the network of computers, right? Nobody send a postcard nowadays. You message or you send a photographs on the social networking sites or the platforms. Similarly, the data that is generated in the field of biotechnology or molecular biology, that can be distributed among the scientific community throughout the world with the help of internet. And again, internet is the network of computers. So we are using computers in biology, in molecular biology, for 4 purposes. First one is storage. Second one is retrieval, that is to get back. Third one is manipulation, that is analysis. And the fourth is distribution. Now, these 4 aspects we fulfill with the help of computer, but we do it for only 3 types of molecule, that is DNA, RNA, and protein. We only talk about DNA, RNA, and proteins in the field of bioinformatics. The reason behind is that only these 3 molecules are information rich. The proteins, the carbohydrates or the lipids, their sequence is just a redundant sequence of the simple monomers. But that is not the case with the help with the DNA and RNA or protein. Their sequence has some meaning to the cell, and that is how the cell functions. And that is why we only talk about only these 3 molecules, DNA, RNA, and protein, because they are information-rich molecules. Now, in bioinformatics, there are 2 subfields. The first subfield is actually the foundation of bioinformatics. As you can see, that subfield include software development, database construction, and curation. Now, when you store the data, you need to have some database system in your computer for the orderly storage of the data. As well as whenever you want to analyze the data, you need to have some software for the analysis purpose. Now, as a biologist, we don't have the knowledge of the programming languages, and we don't usually develop the softwares or databases. So the computer science people, they can develop these softwares, they can develop the databases. So that is the foundation of the bioinformatics. Now, the second subfield of the bioinformatics is applications, where we came into picture, that is the biologist or molecular biologist. We use those softwares, we use those databases for three types of analysis. One is sequence analysis, second is functional analysis, and third one is structural analysis. Now, in sequence analysis, usually we try to compare two gene sequences or two protein sequences, that is called as a sequence alignment. And on the basis of sequence alignment, we can perform gene or promoter prediction or genome comparison or phylogenetic, that is evolutionary analysis of the sequences. And by using that base of the sequence analysis, we can even go further for the functional analysis. Like we can predict the function of the gene, we can predict the function of the protein, or maybe metabolic pathway modeling or protein interaction prediction. Similarly, by using sequence analysis data, we can go for structural analysis like nucleic acid structure prediction or protein structure prediction. That's a part of structural bioinformatics. So we usually work in the field of second subfield, that is application. We use databases, we use softwares which are developed by software developers or database developers, or our own application of these three different analysis. Now, this bioinformatics, it plays a very critical role in all the fields of modern biology. That is proteomics, where you study whole proteome of the cell; genomics, you study whole genome; or pharmacogenomics, cheminformatics, medical informatics, computational biology, or pharmacogenetics; immunoinformatics. I have circled immunoinformatics specifically because nowadays, in the situation of the COVID-19, majority of the scientists, they are analyzing this immunoinformatics data, or the data which is coming from the sequencing, or the sequencing of the coronavirus genome, or our immune system response against that pathogen. So that lot of data is coming, and that is analyzed with the help of these bioinformatics tools. So why do we use bioinformatics? Our first purpose of using bioinformatics is storage and retrieval. Because suppose tomorrow you isolate some bacteria from the soil. Let's take an example that you isolate a bacteria from the soil and you find out that it has some very useful commercial application. How would you identify that bacteria? You can simply go for 16SR DNA sequencing. Now, after sequencing, you can store that sequence into the database. That is the most important function of the bioinformatics. You can store the biological information into the database, as well as you can retrieve, you can get it back whenever you require that information. Then you can compare two gene sequences. Now, suppose you determine the sequence of 16R DNA of that bacteria. Now you want to compare it with the other sequences and find out that how much similarity or how much identity that sequence share with the other sequences, so that you can identify that bacteria at the species or genus level. So that comparison of gene sequences is possible with the help of bioinformatics. Then predict function of unknown genes and proteins. Again, without doing any kind of laboratory work, if you just have a gene sequence or you just have a protein sequence, with the help of bioinformatics tools, we can predict the function of that gene, we can predict the function of that protein. Then we can search for previously known functions of the gene. We can compare our data with other researchers' data, and we can compile the data as well as distribute the data to the other researchers. So if you just submit that sequence to the database, that database will be accessible to all the research community worldwide. And in this way, you can distribute the data to whole community by submitting the data into the database. The next is applications. What are the applications of bioinformatics? Today, everyone is looking for the vaccine of COVID-19, right? So on the right-hand side, you can see this is a structure of spike protein of SARS-CoV-2 virus. Now, in the month of March, this structure of spike protein of SARS-CoV-2 was determined, and it was submitted to the database. It is a PDB, Protein Data Bank, which is the structural database of the proteins. Now, by studying this structure of the spike protein, scientists have find out the mechanism of infection of the SARS-CoV-2 virus. So it is very important for us to understand the structure of any of the important molecule of that infectious virus in order to understand the mechanism of infection. Similarly, in the knowledge-based drug designing, whenever you come to know about the target protein structure, it may be an enzyme, it may be a surface protein, it may be a receptor, anything. If you know the structure of that target protein, then with the help of bioinformatics tool, we can design the molecule which will efficiently fit on that target protein, and it will deactivate it. So that's the whole point of knowledge-based drug designing, where we can use this structural data, structural and bioinformatics data, and we can design the molecule which can actually inhibit the target protein. Next is in the application of forensic DNA analysis. Now, that's not happening in India, but in Western countries, they have created the database of DNA of the criminals. Like in the US and Canada, they have this database called as CODIS, where the VNTR data, that is variable number of tandem repeat data of all the criminals, it is recorded. So there again, this database and bioinformatics tool came into picture, where we can use it for the forensic DNA analysis. Then in case of agricultural biotechnology, now many of the commercially important plants' whole genome has been already sequenced. Now scientists are trying to find out that which genes are responsible for the agronomically important traits, like yield, it may be biotic factors or biotic stress or abiotic stress. What genes are responsible for the response towards that external stimuli? So that is again going on with the help of bioinformatics. Then in molecular phylogenetics. Now again, with the help of sequences, we can establish the evolutionary relationship among the organisms, and that is called as molecular phylogenetics. So this SARS-CoV-2 virus, it is RNA virus, and the size is around 28.9 kilobase pairs. So again, with the help of bioinformatics tools, scientists have come to know that it related to which virus family. By comparing the sequence of this SARS-CoV-2 with rest of the known sequences present in the database, they concluded that this is again a SARS virus, and it belongs to that family. So this evolutionary relationship can also be established with the help of bioinformatics tools. Then we are going to talk about the databases. Now, what is a database? A database is a structured collection of data held in computer storage, especially one that incorporates software to make it accessible in a variety of ways. Any large collection of information is also called as a database. So in the database, the information is stored, or the data is stored in a very structured fashion, so that you can retrieve the data easily from the database. Now, what is database management? The organization and manipulation of data in the database is called as database management. And for the database management, some specialized software package is available, which is called as DBMS, Database Management System. So what is database? Database is a collection of data which is structured. So it is stored in some ordered manner. It is searchable. So you can just simply provide the query to the database, and it will retrieve the whole data related to that query. Like in case of Google, whenever you Google something, actually Google has its own databases of websites. When you type your word or your query into that box, Google will search into the database about that query, and it will show you the results. So database should be searchable. Then it should be updated periodically. New editions should be released on the regular basis, and the database should be cross-reference. Means different databases should be interlinked. They are hyperlinked with each other. Like the protein databases, it is linked with the nucleic acid database, or protein sequence database, it is linked with the structural database. So it should be cross-reference. It includes associated tools also necessary for access of the database, updation of the database, information insertion, and information deletion. And there are variety of database management systems are available, like flat file format is there, relational databases is there, as well as object-oriented databases are there. Now, that is what the database is, and databases are used in a variety of sectors, like in banking sector, in railway booking, or in airplane ticket booking, or even in our colleges also we maintain databases of the students. Now in biology also we have databases, and they are called as biological databases. So what was the need for establishment of the biological databases? In previous few slides, I have explained you that enormous amount of biological data is being produced as the techniques are advancing in the field of molecular biology. As we have these automated sequencers, whole genome sequences are being determined by the scientist. So there is exponential growth in the biological data. And we have to store that data somewhere in the ordered manner. And that is what the need of the biological database is. Now, apart from the sequence data, there is other type of data is also present, that is 3D structures of protein, or two-dimensional gel analysis, or MS, that is mass spectroscopy analysis of the proteins, or microarray data, DNA microarray or protein microarray data is also present. So they are no longer published in conventional manner. So before the age of computer, or before the establishment of the biological databases, whole this lot of data was published in the traditional formats of papers. But nowadays, whole of this data is actually directly submitted to the biological databases, where the whole world can access to this data. Now, essential tools for biological researcher. This database is essential tool for the biological researcher. The only way to publish massive amount of data without using all the paper in the world. Now, earlier to the development of the databases, the sequences were distributed in the form of books or articles from 1968 to 1985. Then from 1982 to 1992, they were distributed in the form of computer tapes. Then came floppy drive. So sequences were distributed in the floppy drive from 1984 to 1990. Then CD-ROM, FTP, that is file transfer protocol, it is being used till date now. Online services, www, this is the most important way of distribution of sequence data, that is World Wide Web, internet, since 1993. And from 2001, we are also using DVDs for the distribution of this sequence data. Some statistics about the biological databases. Roughly more than 1,000 different biological databases are present in the world, and their sizes are also very different. Some databases are less than 100 KB and some databases are more than 20 GB. Like in case of DNA databases, they are more than 20 GB size of that database. Protein, 1 GB, 3D structure, 5 GB, and other databases are relatively smaller. Now, some of the databases, they are updated on the daily basis. So the data is submitted by the researchers on the daily basis, or some are updated annually, and some databases do not update at all. And usually, all these databases, they're accessible through the web. Some are free and some are not free. These are some examples of the databases which we use in the field of molecular biology. Now, categories of database for the life science. Now, as I mentioned, that different types of biological data is being synthesized, and for that different types of biological data, different databases are available. So these are the categories of databases for life science. One is sequence database, where we can store the DNA sequence data or protein sequence data. Then there is a genomics database. There is a mutation and polymorphism database, where only information about the mutation will be stored. Then protein domain and family databases, proteomics databases, where the two-dimensional gel electrophoresis images can be stored, or the mass spectroscopy data can be stored. Then 3D structure of the proteins, they are stored in the PDB. Then metabolic networks, special databases dedicated for that, like KEGG. Then regulatory networks, bibliography. Bibliography in the bibliographical databases, the information about the publications, books, they are stored. Then expression database, dedicated for the microarray data, and some specialized databases are there. So this is the homepage of NCBI, that is National Center for Biotechnology Information. Now this institute was established by NIH, that is National Institute of Health of USA, and a whole lot of work is being done by the NCBI. They have developed variety of databases and variety of computational tools for the researchers in the field of biotechnology and bioinformatics worldwide. So always we heard about the database GenBank. Now GenBank is a nucleotide sequence database which is developed by NCBI, and you can access that GenBank from NCBI website. Also, there is another very interesting tool, that is BLAST, Basic Local Alignment Search Tool. It was also developed at NCBI by Stephen School. So you can access variety of databases from NCBI website, as well as variety of computational tools or softwares from this website. Then this is another very interesting database at NCBI called as PubMed or Medline. It is a bibliographical database. Now this Medline cover field of medicine, nursing, dentistry, veterinary medicine, public health, and preclinical sciences. So all the citations which are published approximately in the 50 to 100 worldwide journals and in 37 language and 60 language for the older journal, they are available in the PubMed database. It contains over 20 million citations since 1948. So if you want to search for a particular research article, you can go to the PubMed website and you just simply type in your query about the research work, and it will show you what sort of publications are already done in that field. So it contains links to biological databases also, and links to some other journals also. And the new data is added in the PubMed on the daily basis. So this is how we can use the PubMed. You simply go to the website of the PubMed. Now, suppose you want to do research on the mitochondria on evolution. So you simply type mitochondria evolution and click on the search button. You will see that how many publications are there on the mitochondria evolution. You can see 3,990 publications were there. And you can simply access those publications by clicking on the link. So it will work similar to your Google. You Google something, it shows you the result, you choose appropriate result out of the list. So similarly, you can do for the PubMed if you want to search for the research article or even for the book. So this is how the abstract looks like when you click on the particular result. You simply type in the query term and click on the go button, and you will get the result. So speaking about biological databases, these biological databases are of three types. That is primary databases, secondary databases, and specialized databases. So we'll talk about them one by one. Now these primary databases contain original biological data, and they are archive of raw sequences or structural data submitted by scientific community. Examples are GenBank and Protein Data Bank. So suppose you have determined a gene sequence and you want to submit it to the databases, you can submit it to only primary database, because primary database accept the raw sequence data. What is raw sequence data? Raw sequence data is unprocessed data. You just determine the sequence, you don't annotate it, you don't describe it, you don't perform the further work about that sequence. Still, you can submit that data into the primary database. So primary database examples are GenBank for nucleotide sequence data and Protein Data Bank for protein structural data. Then comes secondary databases. Now in the secondary databases, a normal researcher cannot submit its entry. because the secondary databases gather the data or take the data from the primary databases. They computationally or manually process it, and after processing, they submit that information into the secondary database. And finally comes third one, specialized databases. Now these specialized databases, they cater to the particular research interest. For example, some scientists, they are working in the field of HIV. Now they are only interested in the DNA sequence, sorry, RNA sequence or protein sequence of the HIV virus. So a dedicated database for them is there, where only HIV RNA sequence and protein sequence or structures are present. Then comes Flybase, where only sequence and structure information of the Drosophila melanogaster is present, or ribosomal database project. They are databases that specialize in a particular organism or particular type of data. Now, these are primary databases. One is, there are three categories of the primary databases. One is for nucleotide. There are three major nucleotide sequence databases in the world. One is GenBank, DDBJ, that is DNA Data Bank of Japan, and EMBL, that is European Molecular Biology Laboratory. Then for protein sequences, there are few databases like SwissProt. Then there is a Tremble, that is translation of EMBL. Then there is a PIR, that is Protein Information Resource. And nowadays very popular one, that is UniProt. Fortunately, for structural data, there is only one database present worldwide, that is PDB, Protein Data Bank. Now, these are all the examples of the primary databases where a researcher can submit the entries directly. So as I mentioned, in case of primary databases, there are three major public sequence databases: GenBank, EMBL, and DDBJ. Now, these three databases, they're available free of cost. Anyone from the world can access these databases free on the internet, and you can download the sequences which are present in those databases. A small number of sequences, especially those published in 1980s, were entered manually from published literature by database management staff. So before the establishment of these databases, whatever sequences are published, they were entered manually by the database staff into the database. Presently, sequence submission is precondition for publication. So as I mentioned that suppose tomorrow you determine some DNA sequence or a protein sequence and you want to publish it into an article, the article will not be accepted by the journal, because for the publication of the sequence data, it is necessary that you submit this sequence to any of the database. If you have a DNA sequence, you have to submit it to GenBank, DDBJ, or EMBL. If you have a protein sequence, you have to submit it to the SwissProt, UniProt, or some sort of database. And after submitting that sequence to the database will provide you with the accession number. And when you have that accession number, then and then only you can submit that data to the publication, or you can publish that work into an article. Now, these three databases, that is DDBJ, EMBL, and GenBank, they collaborate with each other and exchange the data on the daily basis. So earlier to that, the data in these three databases was different. That is, the people who submitted the data to GenBank, it was only available in GenBank. But now this GenBank collaborate with other two databases. So you can see the similar data in all these three databases. And they collaborate by International Nucleotide Sequence Database collaboration. Now, although the three databases all contain same set of raw data, each of the individual database has a slightly different kind of format to represent the data. So this is International Sequence Database collaboration. So if you submit, just take an example, if you submit your sequence to GenBank, what will happen overnight? The GenBank will transfer that file to EMBL as well as DDBJ. So next day, you can see that your data is present in all three databases. That is GenBank also, EMBL also, and DDBJ also. Now these three databases, they have three different search engines present in those databases. For example, in GenBank, Entrée is present. With the help of Entrée, search engine, you can search the whole NCBI and also GenBank. Then in EMBL or in EBI, that is European Bioinformatics Institute, SRS system, that is sequence retrieval system is present. And that works for you as a search engine of EBI. And in case of NIG, that is National Institute of Genetics in Japan, it is Jetenry, where we can use it for the searching DDBJ, that is DNA Data Bank of Japan database. Fortunately, there is only one or single three-dimensional protein structure databases present in the world called as PDB, that is Protein Data Bank. Now, this database archives atomic coordinates. You know, when we determine the structure of the protein, we actually come to know that what are the Cartesian coordinates, that is X, Y, and Z coordinates of each and every atom in the protein or the nucleic acid. And that we do with the help of X-ray crystallography or nuclear magnetic resonance, that is NMR technique. And all these coordinates of that whole molecule, they are stored in the PDB database. The next is secondary databases. Now, sequence annotation information in the primary database is often minimal. Now, before that, let us understand what is annotation. Annotation is a description for something. Like suppose you have a diagram and you write the description about that diagram, that will be called as annotation of the diagram. Similarly, every sequence also have an annotation, that is a description. Suppose you have a protein sequence. Now the annotation for the protein sequence will be which gene is responsible for synthesis or expression of this protein, then what is the function of that protein in the cell, then what kind of post-translational modifications taking place in case of that protein, then what is the subcellular location of that protein. All this information about that protein will be called as annotation of that sequence. So in primary databases, as raw sequence data is also submitted, the annotation level is very minimum. But in case of secondary databases, the sequence which are submitted, they are all annotated sequences. Means the complete information about that sequence you will get in the secondary database. So to run the raw sequence information into, to turn the raw sequence information into more sophisticated biological knowledge, much post-processing of the sequence information is needed. A prominent example is UniProt KB, that is UniProt Knowledge Base, which provide detailed sequence annotation that includes structure, function, protein family assignment, etc., etc. So all the entries in that UniProt databases, they are annotated. Now the gene annotation of each entry is carefully curated by human expert, and thus it is of good quality. So first of all, with the help of computational tools, we can perform the annotation. That is called as computational processing of the sequence. And later on, the human expert also curate that annotation. And that's why the entries which are present in the secondary databases, they are of good quality. As I mentioned, the protein annotation include function, domain structure, catalytic site, cofactor binding, post-translational modification, metabolic pathway information, disease association, and similarity with other sequences. So when you access secondary databases, if you search for a protein sequence, along with that protein sequence, you will also find out all this information about that protein. The examples of secondary databases are ProCite, PFAM, Prodome, Prints, and Blocks. All these are secondary databases for the protein sequences. So what is annotations? Let's take the example of a gene annotation. So how you can perform the annotation of a gene sequence? You have to find out its open reading frames, that is ORFs. You have to find out the functional sites. You have to find out the structure and function. Now suppose this is a gene sequence. Now in this gene sequence, you can see only the repetition of these 4 different nucleotides, A, T, G, C. We do not visualize the annotation or the description of this particular gene by simply reading its sequence. So you have to analyze it with the help of some computational tools and then annotate this sequence and then to describe this sequence. Now, the average length of the gene in the human genome is around 3000 base pair. So I'm talking about the average length of the gene. Now, the gene length could be smaller, it could be larger also, but the average length of the gene is 3000 base pair. Now, in order to analyze this 3000 base pair gene length, you have to use some kind of computational tool, and with that, we can annotate this gene sequence. Now, the annotation of the gene sequence would be like this. The area which is shaded in red, it is a promoter site. Then the sequence that is present before the promoter, that is a TF binding, that is transcription factor binding site. Then the area which is shaded in the green circle, it is a ribosome binding site. And all the codons which are represented in the blue color, they are actually open reading frames. So this is called as annotation, and this annotation can be performed with the help of computational tools developed specifically for the annotation purpose or gene analysis purpose. Then third type of database is specialized database. Now, in specialized database, we usually serve for the specific research community or focus on the particular organism. Like there is a database for model organisms, like in case of Drosophila melanogaster. Only the information of Drosophila melanogaster will be present in that database. The database name is Flybase. Or a specialized database will serve a specific research community. Like I mentioned, suppose people are working on HIV. Then HIV database is a specialized database which only gives them the information about the HIV virus. The content of this database may be sequence or other type of information. And sequences in these databases may overlap with primary database, but may also have new data submitted directly by the authors. Example is Flybase, Wormbase, ASDB, or TAIR. Now in this lecture, we are going to talk about one database, that is GenBank. Now GenBank is nucleic acid sequence database, and NCBI website. You can access this database at the NCBI website. And the content of this database include genomic DNA, mRNA, cDNA, EST, or high throughput raw sequence data, or sequence polymorphism. So a whole lot of DNA and RNA sequence data is present in the GenBank. Now the file that for the one sequence that is present in the GenBank, it has three sections, that is header, feature, and sequence entry. And these three sections, there are many fields. Like in header section, there is accession number or the researcher name or the publication name where the sequence was published. And in the feature section, the whole features or the annotation about the sequence, if available, is present. And in the sequence entry, the actual sequence of that gene or RNA or DNA or RNA will be present. So I will show you the file also. The header section describes the origin of the sequence, that is from which organism the gene is isolated, and then it is sequenced. Identification of organism, then unique identifier, that is the accession number provided to that sequence. Then the top line of the header section is the locus. Locus means location of that sequence in the NCBI database or GenBank database. The identifier is followed by sequence length and molecule type. At present, there are 17 different divisions in the GenBank. Now, these different divisions, they're dedicated for the particular organism group or by using some other criteria. These 17 divisions were set up simply based on convenience of data storage. For example, there is one division called as PLN. Now, in the PLN division, the sequence information of plant, fungal, and algal sequence will be only stored. Then there is a division called as PRI, that is for primates, primate sequence. Then there is a division for MAM, that is for non-primate mammalian sequences. BCT stands for bacterial sequences. Then EST stands for express sequence tag sequences. And next to the division, there is a date when the record was made public. So when you open GenBank, when you search for the particular sequence and you click on it, this type of file will be open. Now you can see at the top, locus is there, and the locus there is mentioned Q9ZGE9. That is actually the accession number of that particular sequence. The length of the sequence is 450 amino acid. It is linear sequence, and it is present in the division called as BCT. That is, it is present in the bacterial division. And when this sequence was published, it was published on 15th June 2002. Then the name of the sequence is present in definition. It is light independent photochlorophyllide reductase subunit N. Again, accession number, DB source, then organism, Heliobacillus mobilis, the whole classification of that organism. Then below that, there is information about the journal where the sequence was published. Then comes feature section. There the in the feature source organism information is there, then gene information is there, then protein information is there, and finally the sequence field is there where whole sequence is written. So that is how this GenBank file looks like, and you can simply download this file into your system, and you can use this DNA sequence or protein sequence for your whole scientific purpose. Now, these are biological databases. Some examples or the symbols I have collected here. Uniprot, Procyte, then Emboss is there, Integro, Sanger, Cath, Scop. All these are different set of databases which are very important in the field of bioinformatics. These are some major databases, as I have mentioned. In case of nucleic acid, there is EMBL, GenBank, and DDBJ. In case of protein, there is PIR, Protein Information Resource, MIPS, SwissProt, Tremble, NRI, and nowadays a very important one is Uniprot KB. Then nucleotide databases. EMBL, it is a nucleic acid sequence database. Ensemble, it is automatic annotation of eukaryotic genome. So suppose you have a genomic sequence of a eukaryote. By using this tool Ensemble, we can do the annotation for that genome sequence. Then Genome Server, it is used for overview of completed genomics, genome sequences at EBI. Then Genome Mod, it is a genome monitoring table. Then EMBL Align, multiple sequence alignment database. So we will talk about the sequence alignment later on. Then Parasite, it is a parasite genome database where the genome sequences of all parasites is stored. Then Mutation, sequence variation database project. So you know that in all individuals, the whole genome sequence is not similar. There is a polymorphism, means at some locations the sequences are different due to mutation. So that sequence variation data is stored in this mutation database. Then IMGT, where this immunogenetic data is stored. And IMGT also comprises that this IMGT and LIGM database. Then protein databases, SwissProt. Now in the SwissProt, you can find most of the entries annotated, means the description for that protein sequence is also present there. Then Tremble, it is the database which is created by translation of DNA sequences present in the EMBL database. Then Interpro, that is an integrated resource for protein families, domains, and functional sites. That is a secondary database. Plus TR, it offers an automatic classification of SwissProt and Tremble database. Similarly, there are many protein sequence databases are there. Now, all these databases which are present, they are correlated with each other or they are linked with each other. For example, suppose we have this genomic data present in the GenBank. Now, for the particular gene sequence file, if the protein sequence or the protein structure is known, then that particular entry will be hyperlinked with the protein database, or it will be hyperlinked with the PDB database. So simply by accessing that single file, you can scroll through all these different databases. So the whole point is all these databases are interlinked with each other, they are correlated with each other, they are not independent. These are some 10 important bioinformatics databases we usually have to work with. One is GenBank for nucleotide sequence. Other one is Ensemble for human or mouse genome sequences. Then PubMed is there. It is a literature reference database, as I have mentioned. Then PIR, Protein Information Resource. It is a protein sequence database. Then SwissProt, again a protein sequence database. Interpro for analysis of protein domains. OMIM is Online Mendelian Inheritance in Man. So all the diseases, genetic disorders which are caused due to single nucleotide polymorphism, that data is stored in this database, OMIM. Then enzyme, again, enzyme data is present in that. PDB, protein data bank, where the protein structures are stored, and cake database. Now in the cake database, metabolic pathway data is stored. So this is the homepage of Entrez Genome, where you can access the whole genome sequences of the organism. Here on the page, you can see Homo sapien genome view. You can access the DNA entries or DNA sequences chromosome wise here by simply clicking on the chromosome. This is the PubMed database, where you can find out the literature data published. This is EMBL database, again a nucleotide sequence database where you can search for the DNA or RNA sequence data. And this is the protein data bank where you can search for the protein structures. After searching, you can see the three-dimensional structure of the protein, and you can also download the file or visualize that protein online. Then again, Ensemble, a genome browser. All the genomic sequence data which is available online, you can analyze it, you can browse it by using this Ensemble database. Next, we are going to talk about the sequence analysis or the sequence alignment. So basically, what is a sequence? Now, what is the sequence? Is the sequence in biology is one-dimensional ordering of monomers covalently linked together to form a biopolymer. Let's take example of a DNA sequence. In the DNA sequence, all these nucleotides, they are covalently linked by phosphodiester bond with each other, and that forms this biopolymer called as deoxyribonucleic acid. Now, this sequence of the DNA or the sequence of the protein, it is also called as primary structure of that molecule. Like in case of primary structure of the protein, the sequence of that protein itself is the primary structure of the protein. And in bioinformatics, we refers to only DNA, RNA, and protein sequences. So what is sequence alignment? Actually, sequence alignment lies at the heart of bioinformatics, because without sequence alignment, we cannot perform functional analysis or structural analysis. First of all, we have to go for the sequence analysis or sequence alignment. So the sequence alignment is a procedure for comparing two or more sequences by searching for series of individual characters or character pattern that are in the same order in the sequence. So suppose you are comparing two gene sequences with each other. What you will do, you will simply find out the matching residue or matching nucleotide among the two sequences. So when you try to find out the matching character or matching nucleotides among the two sequences, that process is called as sequence alignment. Now, two sequences, they are aligned by writing them across the page in the two rows. Identical or similar characters are placed in the same column, and non-identical characters can either be placed in the same column as a mismatch or opposite a gap in the other sequences. Now, in the optimum alignment, non-identical characters and gaps are placed to bring as many identical or similar characters as possible into vertical register, and sequences that can be readily aligned in this manner are said to be similar. So in this example, you can see that if you want to compare these two sequences, two protein sequences, you can see these are amino acids. You simply write them across the page and you find out which residues among these two sequences are matching. Like in this case, L, K, G, K, G, R, D. These residues among these two protein sequences are matching with each other. And at some places, you have placed this gap. You can see we have placed this gap because while comparing the two sequences, we have to remember that these sequences are the product of evolution. And during evolution, mutations occur in the sequence. So whenever mutation occurs, there are three types of mutation. It may be a point mutation, insertion mutation, or deletion mutation. So if in this particular sequence, a residue has been deleted at this position, then you have to place the gap at that place. In this another sequence, if the residue is deleted from the sequence, then we have to place the gap. Now, why these gaps are introduced? In order to increase the alignment score. In order to make this alignment optimum, we have to place the gap. Now, we don't know what happened in the past. During evolution, maybe a nucleotide might be deleted here, or a nucleotide might be inserted here. A nucleotide might be deleted here, or it may be inserted here. So by placing the gap, we increase the chances of the sequence alignment, or we increase the percentage of sequence alignment. That is called as optimum alignment of the sequences. Now, if you find out matching residue among the two sequences, you say that these two sequences are similar with each other. Now, there are two types of sequence alignment. One is the global alignment and other one is the local alignment. Now, in case of global alignment, we compare these two sequences from beginning to end. You start from the first residue and you end at the last residue. And from beginning to end, you align the sequences. You try to find out where the matching residues are present. And in case of second type of alignment called as local alignment, you do not compare the sequence from beginning to end. You just try to find out the local stretches of similarity. Means, suppose actually both sequences are same, you can see in local alignment, we just find out that which residues are matching continuously with each other. We forget about these residues which are present earlier to GKG and after this GKG. We only find out which residues continuously matching with each other among the two sequences. That is called as local alignment. So when to use global alignment and when to use local alignment, let us understand that. In global alignment, we attempt to align the entire sequence using as many characters as possible up to both ends of the sequences. Now, sequences that are quite similar and approximately the same length, they are suitable candidates for the global alignment. Suppose you have two sequences which are 100 amino acid long. Now, those 200 amino acid long sequences can be compared with each other, can be aligned with each other with the help of global alignment. But if you have two sequences, one of the 100 amino acid long, another of 50 amino acid long, now there is a difference of 50 amino acid among two sequences. So you cannot compare those two sequences by global alignment method. Then in that case, you have to perform the local alignment method. For global alignment, one algorithm is developed, which is even used nowadays, called as Needleman and Wunsch algorithm. It produces the global alignment between the pairs of DNA and protein sequences. Next come local alignment. Now, why we have to perform the local alignment? Because during the course of evolution, maybe some sequence have got insertion mutation or some sequences have got deletion mutation, and that is how the length of the sequences got changed. But the functional sites among the two sequences still remain same, because during the course of evolution, the functionally important sites are conserved. They do not undergo mutation. Even if they go undergo mutation, then they will not be selected by natural selection process. And that is why the functional site, it remains conserved. It doesn't change much more in its sequence. But the non-functional sites, they tend to mutate more because they don't play any kind of role in the functioning of that protein or the gene. So when you compare such a sequences where the mutation has occurred, insertion mutation or deletion mutation has occurred, in that case, we have to only see for the functionally important sites or conserved sites. And those conserved sites matching can be found out with the help of local sequence alignment. In the local sequence alignment, we only find out local stretches of sequence similarity with the high density of match. Then it generates one or more islands of matches or sub-alignment in the aligned sequences. Like in previous example, we see that in local alignment, we only find out this island of matching residues among these two sequences. Well, when we perform global alignment, we compare two sequences from beginning to end. But when we perform local alignment, we only find out the maximum density of matching residues among the two sequences. Because most probably, this is the site which is functionally important, and that is why it is conserved among both of these sequences. So suitable for aligning sequences, this local alignment is suitable for the aligning sequences that are similar along some of their length, but dissimilar in others, and sequences that differ in the length or sequences that share conserved region or domain. There is another algorithm for local alignment called as Smith and Waterman algorithm that is used to perform the local alignment of DNA and protein sequences. Then methods of sequence alignment. Now there are three methods of sequence alignment. One is dot matrix method, where we simply qualitatively find out the matching residues among the two sequences. The second method is dynamic programming method. Now this dynamic programming method is a mathematical method of sequence alignment, and which is most accurate. It produces 100% accurate alignment among the two sequences. And the third method is word method, which is a heuristic method. So usually it takes the shortcut and it gives you the result in the real time frame. For example, BLAST and FLAST. So what this word method does, this word method compare the sequences in the form of words, and then it shows you the alignment score. And that is why it is a shortcut method. But the application of this using word method is it is very simple, and it can be done in a real time frame. Means when you perform the BLAST or FASTA analysis, it gives you the result within minutes. BLAST and FASTA, these are two tools which are used for the database similarity searching. Now in the database similarity searching, you provide a query sequence to the database, and database will compare, or these two tools will compare the query sequence with all the sequences present in the database. So suppose database consists of 1 lakh sequences, and you provide one query sequence to compare with the database. Means this BLAST and FASTA, they have to perform the sequence alignment 1 lakh time. That is your query sequence and 1 lakh sequences from the databases. They have to align them all. So when you have to align so many sequences at that time, some shortcut method will be very useful, because it will save you the time, it will save you the computational power, which dynamic programming cannot do. The dynamic programming will take hours to search the matching sequence from the database, where this word method, that is heuristic methods, BLAST and FASTA, they will provide you the result within a few minutes. So that is all from my side. If you have any question, you can ask.
Yes, now we'll take a question and answer session. I would like to request Professor Saraswati, please hand over.
Okay, sir. First question, what do the positive and negative symbol mean on blast sequence alignment results?
What are the
What do the positive and negative symbols mean on blast sequence alignment results?
positive and negative symbols?
Okay.
Okay, so when you perform the alignment, actually, BLAST matches your query sequence with all the sequences present in the database. Now, wherever there is a match among the residues, it will show you with the positive sign. And wherever there is a mismatch, it will show you with the dash or the negative sign. So that you can simply interpret it as matching and mismatching of the residues. That usually it does with the DNA sequences, but with the protein sequences, in the middle line, it simply write the name of that residue. If there is a match, it will write the name of that residue. And if there is a mismatch, there will be nothing.
Okay, sir. Thank you. So next question, why the sequencing peaks for the same nucleotide base in picture differ in their heights?
In sequencing, could you please repeat the question?
Okay. Why the sequencing peaks for the same nucleotide base differ in their heights?
Okay, so that's the height is actually referring to the intensity of the light which is emitted by the fluorophore which is attached to that nucleotide. Are you talking about the pyro sequencing or Sanger sequencing? In case of Sanger sequencing, nowadays, 4 different nucleotides, that DD-NTPs, they are attached with the 4 different fluorophores. So the laser light is continuously scanning the nucleotide, and the nucleotide fluorophore got excited and it will emit the light. And that light will be detected by the detector. So on the basis of that intensity of the light, the height of the peak will be different. The height of the peak in case of Sanger sequencing is not important. We only have to consider the color of the light, because that color will tell us that which nucleotide it is. And if you're talking about the pyro sequencing, then in case of pyro sequencing, if the height is twice as much the height of the normal peak, then it indicates that 2 nucleotides are present of the same sequence. If the height is of thrice, then 3 nucleotides of the same sequence are present. So the meaning of height is different in Sanger and pyro sequencing technique.
Okay, thank you, sir. So next question, is this useful for medical research? Bioinformatics is useful for medical research?
Yes, of course, it is useful for the medical research. Like I have mentioned in my presentation also, there is one dedicated database for that, that is called as OMIM, Online Mendelian Inheritance in Man. So all these new genetic disorders that we are finding out in the humans, all that polymorphism data is stored in that OMIM database. And another use in the medical research, we can say, is a knowledge-based drug discovery. Like in the pharmaceutical company earlier, what they were doing, they had this chemical database. Means they actually synthesize the chemicals, which are novel chemicals, which can be used as a drug molecules, and then they screen all those chemical molecules, the compound molecules against the target protein. But nowadays, with the help of bioinformatics, what we can do, we can use the docking softwares for the virtual screening of the molecule. So suppose you have a virtual library of the chemical compounds and you have a structure of the target protein. So you can virtually screen that whole chemical library. It consists of thousands or millions of molecules against the target protein, and it will give you maybe some few hundred hits which you can analyze in the laboratory. So in the drug discovery also, it has a huge application.
Okay, thank you, sir. So, sir, next question. Is any sequence alignment done between coronavirus and rhinovirus for phylogenetic analysis?
Yeah, scientists are doing that for phylogenetic analysis. Not only rhinovirus, you can do it with other RNA viruses also.
Okay, sir. Thank you.
Mhm.
So much, sir. Now, I'd like to request the teacher, Professor Anton, to give the voice for
Uh, it is my great privilege to express my sincere vote of thanks to all who have helped us in making this national webinar on bioinformatics concept and development. First of all, I would like to propose heartily vote of thanks to our chief person, Honorable Shri Balasaheb Wagh, President KK Wagh Education Society, for gracing today's webinar. I would like to thank our today's resource person, Mr. Ashish Wani, for making excellent presentation and making this webinar interesting and meaningful. I would like to express my sincere thanks to all trustees and secretaries of KK Wagh Education Society for their blessing to this online webinar. I would also like to thank Dr. V.M. Sevlikar sir, Campus Coordinator, KK Wagh Education Campus, Saraswati Nagar, Nashik. I would like to express our profound gratitude to our principal, Professor N.S. Pastor sir, for his guidance to make this webinar session successful. I am happy to express vote of thanks to all the dignitaries, principal, HOD, staff members, and students for the participation and making this event successful. Once again, I thank all of you for your coordination. Thank you.
the wonderful explanation about bioinformatics. Thank you so much. And
Thank you, sir.
all, I would like to request all participants that the feedback link and the e-certificate is already shared in the chat box. You can go through the chat box and please give the feedback link. Thank you so much. And with the prior permission here, I announce that today's webinar session is over. Thank you so much, everyone. Thank you.